leadership.
Okay, you guys. So I need you to get two things out. Get your dihybrid problems as well as your notes. So get those two things out. Did you write like how many, like 9, whatever? Like, what do you mean? Like, did you write it out like there's 9?
Like all those.
Or did you just do the punishment? So in your explanation, did you say that you have so many of one kind and so many?
I think he was saying like, do we have to do the Punnett squares, or could we have just said like there's 9?
No, he was saying that he didn't do the ratio, but he explained it more.
Oh yeah, that's what.
Yeah. All right, that's fine. Okay, in just a minute, I am going to collect these, but not until a little bit later in the hour, because I'm going to teach you some things of how to complete number 6 and 7 that I had you skip. All right, so we're going to be doing that as we go into the hour. But yesterday, I did upload the answers to the problems, so that if you ran into trouble, you could check your answer and therefore see, so you could kind of maybe figure it out, and it would help you with the other problems. But what I'll do is just quickly go over, I'll just quickly just say the answer, so if you didn't get a chance to do that, you know what the answers were. So for number 1A, all the kids should be both heterozygous, all heterozygous, so big W, big H, little h. And for B, for number 1, your ratio should be a 9:3 to 3:1 ratio. Number 2, you should have gotten at the end a 4:4:4:4 ratio, or if you reduced it to a 1:1:1:1 ratio, that's the same thing. Number 3A, you should have gotten that all the kids are heterozygous, big B, big S, little s. And for B, it was also a 9:3 to 3:1 ratio. Number 4, you should have gotten a 4:4:4:4 ratio, or 1:1:1:1. And then lastly, number 5, you should have gotten 3/4 of your chances of having a child that would be deaf. All right, so I had this up on the website. So anybody have any questions or want me to go over a particular problem? Sometimes you can look at the answers and kind of figure it out what you did wrong from there. So, but if you can't figure it out, I wanted to give you an opportunity to ask. We good? Okay. All right. Perfect. So I'm going to have you hold on to it, and as I said, we're going to go on in our notes. I'm going to teach you a few new things, and then we'll come back to number 6 and 7. After we do that, then I'll collect it, okay? And I'll give it back to you tomorrow so you have them to study from for the test, okay? So here we are. This is where we left off in our notes. So we're on 14.2. All right, so Mendel's law of segregation and independent assortment reflect what we call the rules of probability. And so I'm going to teach you a few things about these rules of probability. There are 2 of them. So they give an analogy with tossing a coin. And every time you toss a coin, you have a 1/2 or 50% chance of getting heads, or 1/2 or 50% chance of getting tails. And when you flip the coin once, that probability, 50-50 heads or tails, doesn't affect the next time you flip the coin. So the next time you flip the coin, you still have the same probability of 50-50. And so that's what this is showing you here. And so this relates to genetics in the same way the alleles of one gene segregate into gametes independently of another gene's alleles, meaning that if you have alleles that are on different chromosome pairs, so here's one chromosome pair, let's say the individual's big A, little a for a gene on that chromosome pair, and then they're big B, little b on the second chromosome pair. So what happens is 50% chance of, for this particular individual, giving a big A or a little a to their kids, because when they, if this is their genotype, when they make their gametes, they're going to make half their gametes with a big A, half their gametes with a little a. And that's independent of the second chromosome pair. So this chromosome pair also, the 50% of the gametes at the end of meiosis will have a big B, and 50% will have a little b. And so how these guys line up, this pair and this pair, everything's independent of the other pair, all right? And so that's what this means. So the two rules with probability are the rule of multiplication and the rule of addition. So the multiplication rule states that the probability that two or more independent events will occur together is the product of their individual probabilities. So we take the individual probabilities and we multiply them together. So oftentimes we're looking at, in this case, you see the word and, like, what's the probability of this and this occurring together, okay? And so that's when we would use the multiplication rule. So an example, having a boy and then a girl. So when you have a child, you have a 50% chance of having a boy and a 50% chance of having a girl, a 1/2 chance. The reason why, remember that males, when they're going through meiosis, that 23rd pair, they have an X and a Y chromosome. And so going through meiosis, pairs of chromosomes segregate into different cells. And so when males make their sperm cells, they make half with a X chromosome, half with a Y. Females make all of their egg cells with an X because they have two X chromosomes, so when they segregate and go into opposite cells during meiosis, all of the cells have X chromosomes. So that's why, so it depends on if the X sperm or the Y sperm fertilizes the egg. So having a boy the first time you have a child, there's a 1 out of 2 chance. And then you have another child, the chance that child's going to be a girl is 1 out of 2 chance. And so then what we do with those probabilities, what's the probability of having that boy and then the girl in that order, we take the individual probabilities and we multiply them together. What's the chance of those two occurring together right after each other? So we take these and multiply them, and so you have a 1/4 chance of, when you have 2 kids, of it being in that particular order. So going with that same idea, flipping on a coin, heads, then tails. So you flip 3 times. What's your probability of flipping 3 times and it being heads, tails? What do we do? Multiply. So what's my answer going to be when I multiply this together? 1/2 times 1/2 times 1/2 gives us 1/8. All right. So that is your answer there. All right. So that's the rule of multiplication. And so we're going to apply this to some crosses here in genetics in just a second. So the probability in an F1 monohybrid cross can be determined using the multiplication rule, and I'll model that and show you that in just a minute here. So segregation in a heterozygous plant is like flipping a coin. Each gamete has half a chance of carrying the dominant allele and half a chance of carrying the recessive allele. And so what does that mean? So if the individual's heterozygous, they're big A, little a, when they make their gametes, half their gametes will have a big A, half will have a little a. So that's your half and half chance of the dominant and the recessive allele. So this picture here is showing you a genetics problem and using it with an analogy with flipping a coin. And so, so when this is your cross, big R, little r, cross with big R, little r. So we're looking at 2 heterozygous parents. And so remember when they're heterozygous, I'll just change these to R's. They're big R, little r on a chromosome pair. And so they have, as we learned before, they make 2 kinds of gametes. They either pass on the big R or the little r, kind of like just flipping a coin. Flipping a coin, you're going to get heads or tails. It's 50-50. Same thing with making the gametes, so that's why we have the big R and the little r here. And they put the half in front of it, meaning that's the probability that this this is the half their in this case sperm will be a big R, half will carry a little r. And then the other parent, they're both the same in terms of the genotype, so we have to set up the same. So then when you fill in the Punnett square, we've already done this and talked about probability when you look at the Punnett square. So when you're looking at this, what's the probability of having these two parents having a big R kid? So if you were to give that cross and figure out the probability of having a big R kid, you could make the Punnett square and say, oh, there's one box out of the 4 boxes that is big R, so that's a 1 out of 4 chance, and that's my answer. So that's where they have here, 1 out of 4. But you can also use the rule of multiplication. So to get a big R kid, that means that each parent had to give a big R. So this parent, the chance of giving it a big R is 1/2. This parent, the chance of giving a big R is 1/2. So the chance that those two are going to come together and have a big R individual would be 1/2 times 1/2. And that would also give you 1/4 is the rule of multiplication. So when looking at this, you could, if you look at this cross without doing the Punnett square, and you were asked what's the chance of these guys having a big R kid, in your head you could say, okay, this has a 1/2 chance of giving a big R. This one has a 1/2 chance of getting a big R, so 1/2 times 1/2 is 1/4 without even doing the Punnett square. Does that make sense? Okay? All right, so that's the rule of multiplication in this case. Let's talk about how you figure out big R, little r. So if you do the Punnett square, hopefully most people would be able to tell me that there's 2 out of the 4 boxes that show a big R, little r, so that's 2/4 or 1/2 or 50% chance that these parents can have a big R, little r. But let's talk about the math here. So look at here, there's 2 different ways that you can these two parents can have a big R, little r. There's 2 different ways that can happen. This parent can give the little r, and this parent give the big R, or this parent give the big R, and this parent give the little r. So there's 2 different ways that can happen. And there's a 1/4 chance of it happening this way, and a 1/4 chance of happening it this way. So what you do in this case, if, without doing the Punnett square, is we don't use the rule of multiplication. We use, next thing, the rule of addition. The rule of addition states that the probability that any one of 2 or more exclusive events will occur is calculated by adding together their individual probabilities. In this case, a lot of times you have the mindset, and it might even be better in the stem of the question, or. So if there's 2 ways that something can occur, so it could occur this way or this way, what's the chance of that happening? We add the individual probabilities together. So if we go back and apply this to my example with the big R, little r, so if I ask these 2 parents what's the chance of having a big R, little r kid, it can happen this way or this way. So the chance of it happening this way is 1/4. The chance of it happening this way is 1/4. So therefore, the chance of it all together happening is 1/4. Right. 1/4. What do we do with a 1/4? We add them together, and that gives you 2 out of 4 or 1/2. And it's the same answer there. All right. So if I were to ask you, what's the chance if you roll a die, a regular 6-sided die, what's the chance of rolling a 1 or a 6? Yeah, it is 1/3, and the reason why is that every time you roll, you have a 1 out of 6 chance. I gotta get a different color. 1 out of 6 chance of getting a 1, and you also have a 1 out of 6 chance of getting a 6. And so, what's your chance of one or the other? Then we take those individual probabilities and add them together, which is 2/6, which is 1/3. All right, and so that's another application of that. That's the rule of addition. So the rule of addition can be used to figure out the probability that an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. And I'm going to go through some examples of these in just a second. And as I said, it often uses the word or. So applying these 2 rules to genetics, solving complex genetic problems with the rules of probability, we can apply multiplication and addition rules to predict the outcome of crosses involving multiple characters or multiple traits. That's like the dihybrids. So this is where we're going to come back to the dihybrids here. A dihybrid or other multi-character cross is equivalent to 2 or more independent monohybrid crosses occurring simultaneously at the same time. So, so it's saying here, if you're looking at 2 traits, so one trait, the R's, and then another chromosome, another trait. So, and crossing it with another parent that has the R's and the A's, you can think of it as a dihybrid cross, or you can look at it as 2 monohybrid crosses occurring at the same time. I'm going to show you an example of that in just a second. So, in calculating the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied together. So let me explain or give you an example of that occurring. So let's go back to your dihybrid crosses, and I'm going to use the example with number 2. Number 2, you can make a big Punnett square, or you could look at them, as this shows you, the last 2 bullets, as 2 separate monohybrid crosses. So if we look at number 2, and let me put the answer up here. So this was your cross for number 2, and this is how you set up the large Punnett square, the 4x4 Punnett square. And we got the 4 to 4 to 4 to 4 ratio there. What I'm going to do is show you a different way to do this. And so I'm going to rewrite, so it's big W, little h, little h. So I'm going to rewrite this so I can show you a separate way. So this is one parent, and the other parent was little w, big H, little h. And so what you can do, what this is saying here, is that yes, this is a dihybrid cross. We're looking at 2 individuals and we're focusing on 2 traits at once. But these 2 traits, since they're on 2 different chromosomes, can be considered as 2 different monohybrid crosses. So what you can do is focus on just the W's first, all right, the 1 chromosome pair. So right now we're going to just look at these 2 parents and disregard the H's for a second and just do a monohybrid cross looking at just the W trait. And I can set up my Punnett square, so this parent could give a big W or little w. This parent is both little. And so this is my possible combinations in my kids. And so I have a half, these parents have a half of a chance of having a kid that's big W, little w, and a half that's little w, little w. And so that's just looking at the W traits. Then I can look at the H's. So looking at H and H. This parent is always going to give a little h. This parent could give the big H or the little h. So when I'm filling this out, these are my possible combinations, and I have a half a chance of, these parents have a half a chance of having a kid that's big H, little h, a half a chance of having a kid that's little h, little h. All right, so those are my 2 traits. So what we want to do then is figure out, like for instance, what the probability of these occurring together. So these 2 parents have a half a chance of having a big W, little w, and a half chance of having a big H, little h kid. So what are my chances of having both of those traits in the same individual? Big W, little w, big H, little h. What do I do with those individual probabilities? I multiply them. So therefore 1/2 times 1/2 would give me 1/4, and so I have a 1/4 chance of having a kid like this. I could also have the big W, little w kid with what other combination? Little h, little h. I could have this combination here. So big W, little h, little h. 1/2 times 1/2, I have a 1/4 possible combination there. And then that's it for the big W. So then I could add, I have a half a chance of having a little w kid, and then another half chance of having a big H, little h. So those 2 occurring together, we take those and multiply them together. So we have little w, big H, little h. And then what's my last combination? All recessives. So then I have a 1 out of 4 by multiplying these together to get all recessives. And notice here that This is kind of like, this is showing both the dominant traits. This is showing one dominant, one recessive and one dominant, and all recessive. And this is like a 1-to-1 to 1-to-1 ratio, which when you did the big Punnett square, you got the same ratio, all right? And so therefore, you can get the same answer by doing a large Punnett square or two individual smaller Punnett squares and using the rule of multiplication. All right, what I'd like you to do is take a look at number 6 in the dihybrid problems. Take a look at number 6. I'm going to throw a little curveball at you. Number 6 has 3 gene pairs, the K's, the L's, and the M's. So I want you to use the strategy that we just went over with this problem and looking at each gene as a separate monohybrid cross. And look at the question that's asking you, what's the chance of these two parents, and they give you the genotypes of the parents, of having, I think this one asks you for an all recessive kid. So therefore, what I'd like you to do is use the individual Punnett squares to see if you can figure out that chance. It's much easier to look at the individual Punnett squares than it is to make a huge, big Punnett square for 3. Take my word for it. test, and it's asking you about what's the chance of all recessive. So if you do the different Punnett squares, so for the K's, when you do that, as a 1/4, there's a 1/4 chance of having a little K, these parents having a little K kid. If you do the L's, there's a 1/4 chance of little L, and you do the M's, there's a 1/4 chance of little m. So therefore, what's the chance of all of those occurring together in the same individual? You take those individual probabilities and you multiply them together, and you should have 1 out of 64. All right, and so. So if you haven't already done so, I'm going to have you do number 7. Number 7 throws five genes at you. All right, so 5 gene pairs, I should say. Okay, so they gave you the cross, this cross here, and asked you what's the chance of having a certain kid, and the answer is 1 out of 512 is the answer. All right. So how do we get that answer? So when you look at the D's and cross the D's, so you have a little d kid, the probability of that is 1/4. So let's look at what we're multiplying together. The probability of them having a little big E, a kid is 1/4. Big F, little f is 1/2 or 2/4. Little g is 1/4, and big H is 1/4. So we multiply those all together, and we get 1 out of 512. If you left the F says 2/4, you got 2 out of 1,024. But if you divide that, reduce that, it's 1 out of 512. Yes.
Is it okay if we don't like write all this out, like every single one of the Punnett squares we need to do on this?
No, it's okay. If you get to a point where you can get in your head like what the chances are, you don't necessarily have to draw the Punnett square out. Okay, so like if this was one of the math problems on the AP test, like what's the probability of this, and put your answer in a fraction. So you could just, if you don't need to draw the Punnett squares out, it can save you some time. If you can't do it in your head, please do write out the Punnett square so you can make sure that you do it correctly. All right, but you don't have to. So that's kind of what I did. I could kind of look at it and tell. All right, so absolutely. Okay, so I'm going to collect the dihybrid problems. So make sure your name is on them. If you put it on the separate sheet of paper, I don't really care when you pass them in if you have the question sheet attached to it, unless you care. If you have multiple sheets, you might want to staple them together. Like if you did 6 and 7 on a different sheet or something like that, you can staple them together. Or if you want the question sheet attached to it, you can staple it together. But let's pass it forward to the front of the room.
Yeah.
Okay, to be just, okay, so all the ones with the big answers. And then, but they have to have a big K for here. So therefore, I'm gonna tell you about that. Also, the absence of the K. Awesome. Okay, so that's probably right. Thank you. You're welcome. Okay. So now if you go back to your notes, there's a Punnett square, and I put the boxes, I drew the boxes in there like I did with the one yesterday from the notes, so it's a 5 by 5 square, so you have to put the possible parents. And so what I'd like you to do is to do that. It gives you the parents in a cross, and it asks you what's the chance that they will have, the kids will have, and it says any 2 recessive genes. And so what I was trying to get at here is that and the recessive gene could be from the same gene pair or not. So it could be a little b and a little r. And it should be at least any 2. So it could be more than 2 recessive genes. So go ahead and do that and see what you get. And I'm going to get your dihybrid problems. R, little r. So therefore, if little r, they show
Sage
up.
reminds
So
us of the year.
that's where it is.
The what?
Lesson of the year. You don't.
Okay.
Oh, I do know. Nick knows me that I am, but I'm like,
No. It's not a goal.
Yeah.
your bees start to look like green Vedas. Unless you pass on
You get 50%.
Did I ask you both? Can they be like a recessive ruby and a recessive? Like, would this count?
So, yes, so this has, any two, so it doesn't have to be together.
Yeah, like this one?
Like, we,
That one.
it doesn't have to be
Two.
So when you're setting this up, so you're setting this up. This first parent, there's 4 combinations to pass in their eggs or sperm cells. The big B and the big R, the big B and the little r, little b and big R, little b and little r. So that's one parent. The second parent is big B, little r, little r. So all their gametes are going to have a big B and a little r. So notice what I did is that I wrote big B and little r, but that's going to be the same for each one of these because that's the only combination that that parent could give. And so therefore, when you're doing a Punnett square, you could technically just set it up like this and you get the same ratio and you can save yourself a lot of time, all right, with that, because these rows essentially are going to be the exact same as these rows multiplied by 4. There's just 4 times as many of them, all right, and so so that's a little hint to help you to save time also. And so then any 2 recessive genes, at least any 2 recessive genes, would be these 3 here, which would give you 3/4. All right, and so you have a 1/4 chance of this, a 1/4 chance of this, a 1/4 chance of this. So I just applied the rule of addition here. So this plus this plus this gives you 3/4 as well. All right, because it can happen in one of those 3 different ways. All right. All right, so we're gonna go on in our notes. So let's move on to 14.3. Okay, so the relationship between genotype and phenotype isn't always as simple as what we've been talking about with Mendel and his pea plants. So we're gonna look at some different scenarios. Many heritable characters, that means traits that are inherited, are determined by only, are not determined by only one gene with two alleles. Like, for instance, pea plants with flower color. There you have a big R is purple and a little r is white, and all pea plants have some combination of those two, and purple is dominant to white. There's a lot of genes that there's more to it than that. However, what Mendel came up with about how the laws of segregation, how the chromosomes segregate or separate into opposite cells, and you only give one to each, each parent only gives one to their kids, is still the same. So, inheritance of characters by a single gene may deviate from those simple Mendelian patterns in these following situations. And we've already talked about this first one. The first one, it says when alleles are not completely dominant or recessive. Do you guys remember the scenario that we've already talked about with when alleles are not completely dominant or recessive? Remember, that was the red and the white flowers, and if you get a red and a white gene, instead of appearing red or white, those heterozygous individuals appear pink, and you got that blending. That's called incomplete dominance, and we'll review that here in a second. The second scenario that doesn't follow what Mendel came up with, that we've learned later, is when a gene has more than two alleles. Like flower color, there are only two alleles, purple and white, and every flower has a combination of those two. Some genes have more than two alleles. And then when a gene produces multiple phenotypes, and so we'll look at that as another example as well. So the spectrum of dominance, this is what we've already talked about. So we've already done this slide here with complete dominance, which is like Mendel's pea plants with flower color. Purple is completely dominant over white, so that when you have a heterozygous individual, you only see the dominant trait. Codominance, I kind of glossed over this. We wrote about it and I said it, but we didn't really talk about it. And so I'm going to put here an example is type AB blood. We're going to talk about that here today. This shows codominance. We'll get to that in a second. And then incomplete dominance is where we get that blending that we talked about earlier with the red and the white. And so this here shows incomplete dominance. So incomplete dominance is a blending. You see the mixture there of the heterozygous individuals. Red is not dominant over white, and vice versa. So when they both show up, they blend together and we get that pink color. So a dominant allele does not subdue a recessive allele, and alleles don't interact. Alleles are simply variations of a gene's nucleotide sequence. And so here, when you have the purple gene versus the white gene, what makes them different is the order of the bases, A's, T's, C's, and G's, and the DNA that codes for that gene. So for any character or trait, dominance or recessiveness relationships of alleles depends on the level in which we examine the phenotype. And so we're going to look at these different phenotypes. So yesterday I kind of talked a little bit about this already, the frequency of dominant alleles, that dominant alleles are not necessarily more common in populations than recessive alleles. The trait I use as an example of that is what? Yeah, the 5-finger trait, and having more than 5 fingers or toes or more than 5 digits on a hand or a foot, right? To be specific. So for example, one baby out of 400 in the United States is born with this condition. It's called polydactyly. Most people do not have this condition. So the allele for this unusual trait is dominant to the allele for the more common trait of 5 digits per appendage. So in this example, the recessive allele is far more prevalent than the dominant allele in the population, and that's why the frequency of people getting that dominant allele is so low, because most people are little r in the population. All right, so some traits exhibit what we call multiple alleles, and our human blood type, this is the case. So most genes exist in population in more than 2 allelic forms, meaning that there's more than 2 alleles to choose from to make up a person's genotype. So for example, the 4 phenotypes of the ABO human blood group in humans are determined by 3 alleles for a particular enzyme that we're going to say is the capital letter I, that attaches either A, carbohydrates, or B, carbohydrates to the surface, outside surface of our blood cells. And so we have 3 different alleles in humans for this to determine your blood type, AB, or O. So this big IA, I'm going to put here in the margin, big IA is, stands for A blood. People that have this big IA gene, that big IA means that they make an enzyme that'll attach A carbohydrates to the surface of their red blood cells. A big IB gene means that they make another enzyme that attaches B carbohydrates to the red blood cells. So big IB will give you B blood. And then we have little I will give you O blood. And and so little I, we're going to see here next, doesn't attach any carbohydrates. So the enzyme encoded by the big IA allele adds the A carbohydrate, whereas the enzyme encoded by the big IB allele is the B carbohydrate. The enzyme encoded by the little I allele adds neither. And so, so it doesn't add either one of those carbohydrates to the surface of the red blood cell. Let's look at the way that these are written. This one is little I versus 2 big I's here. What does that mean? What can you deduce how the little I gene compared to the other 2 genes?
Well, the first 2 and B are both dominant. Well, they're both dominant.
So they're both, these guys are dominant to what?
Dominant
To the little I. All right, absolutely. Yes.
When you say AB carbohydrates, these are separate from the antigens that we talked about previously.
No, these are the antigens.
These are,
Yes,
okay.
yes, absolutely. So these, when we talk about A and B carbohydrates being added to that, when we talked about A and B blood and so on with the immune system, those are the antigens. They're just carbohydrates. They have a certain shape that act as your antigen. All right. All right, so in this particular chart here, let's go through the 4 possibilities of your blood type. So we're not going to talk in this about the Rh factor, the positive or negative. So when we're looking at these A, B, and O, notice here I have a genotype here. And so if you have A blood, you have 2 genes, one that you got from mom, one that you got from dad, that determines your genotype. And for the human population, there's 3 different alleles that everybody's genotype is made from. So some of us in here are going to be little I, some of us will be big IA, little I, some will be big IA, and so on. So we have 3 alleles to choose from. So if you have A blood, what do you think is true? What has to be true about your genotype? What do you have, do you think? You have to have a big IA. So you have to have an A gene. Now, what could your second gene be? It could be another big IA gene. So you have 2 A genes, and they'll for sure give you A blood. But I heard some people say what? That could also be what? A little I. So it could be this or big IA with a little I, because that A gene is dominant to the O gene, and so you would make that enzyme and add the A to the surface of your red blood cell. So if this is your red blood cell, you would add these A, all right, add them onto the surface of your red blood cell, because you have the big IA that makes an enzyme to do that. For B people, what do you think? They have B blood, so these guys have to have a B gene. They could also have a second B gene, so they could be big IB, big IB. These guys would be homozygous, all right? They'd both be B genes, or they have a little I with that. Either one, they have B blood, and so their surface of the red blood cells are like this, all right? So the difference of these are B because they make these genes make code for making an enzyme that attaches those homes. All right, AB, what do you think of AB?
Able
These individuals have both an A gene and a B gene. So these guys are big I A and big I B. And that's the only combination that they can be. There's no other combination. This is where codominance comes into play. So I said earlier we wrote example type AB blood. Why this is codominance and how it differs than incomplete dominance. Codominance means that neither one of these are dominant over the other, but they both make their enzyme. So this gene codes in your body makes the enzyme to add the A to the surface of the red blood cell. This enzyme also this gene also works to make enzymes that add the B. And so therefore, this is what the surface looks like for people who have type AB blood. They have the A and B attached to it. And so it's not a blending. So there's not some weird intermediate between the A and B. That's incomplete dominance with the pink. This is codominance, all right? And so therefore, that's that. And then O has, yeah, O's have little i's and they don't have, their surface is nothing on there. So you guys remember this a little bit?
Yeah.
So I have a question about like the codon.
Yeah.
So like when you have a dog that has like 3 different colored fur, does that mean they have 3 different genes with 3 different colors?
Yeah, there's a lot that goes on in that. But yeah, there's genes that determine the color. So there's different genes for the different colors, but there's also genes about depositing those colors and where and if and what, like those genes. So it's very complex. All right, but yes, but you have different genes working all together to ultimately control that. Yeah, yeah. Question? So, yes. So this is recessive. So the little I is recessive compared to either the big IA or the big IB, which is why when you have a big IA and a little I, they have A blood because the A is dominant to the O. All right, absolutely. All right, so what I'm going to have you guys do is, I think you know enough now to what I'd like you to do is do these, just the front side of this. Don't worry about the back side right now. The front side of this are blood type problems. And so I give you information about 2 people. I give you enough information. I tell you if they're heterozygous, homozygous, and so on, that you should be able to figure out the genotypes and do the problems. Okay? So, yeah, for this, I'm going to have you do that. All right.